Hey there, Dr. Avinash here. This is a quick video for helping you revise for your NEET 2026. What you need to remember is as follows. First, very odd question that can come is Alfonso Corti. This guy was an Italian anatomist, and his main contribution was his study on mammalian auditory system. In 1851, this guy published a paper which described the structure, which was located on a membrane called as the basilar membrane. This is in our cochlea. This special structure contained hair cells, and these hair cells were responsible for converting sound vibrations into nerve impulse. This structure was called as the organ of Corti or organ of Corti. So you just have to know this much. Okay? This was extra. Let's go on to our main chapter, breathing and exchange of gases. These are the following things that you must remember. Understand. Oxygen is utilized by organisms to indirectly break down simple molecules like glucose, amino acid, fatty acids to derive energy. But as a result of this breakdown, you have carbon dioxide produced. Right? This carbon dioxide has to be thrown out. So, what we have established is oxygen has to be continuously provided to the cells and carbon dioxide has to be released out. This process of exchange of oxygen from atmosphere with carbon dioxide produced by the cell is called breathing, also called as respiration. Easy? Cool. Now, the mechanism of breathing will vary. Different animals will have different types of mechanisms. MCQ. What does this mechanism of breathing depend on? Depends mainly on their habitat and level of organization. Let's take some examples. Invertebrates like sponges, coelentrates, flatworms. They exchange O2 with CO2 by simple diffusion over their body surface. They do not have any special respiratory organs. Okay? Earthworms use their moist cuticle. Insects have a network of tubes called tracheal tubes, which you probably studied in cockroach chapter. Apart from this, you would notice that there are some aquatic arthropods and of course mollusks which have specialized vascularized structure called gills. MCQ. In our textbook, gills are described as branchial respiration. So, aquatic arthropods and mollusks use gills. Whereas, the terrestrial forms of the above animals, terrestrial arthropods, terrestrial mollusks, may have vascularized bag called lungs. Lungs are described as pulmonary respiration. Vertebrates are different. Vertebrates, fish use gills. Amphibians, reptiles, birds, mammals have lungs, and apart from this, amphibians also use their skin. And if you remember, amphibians also use their buccopharyngeal membrane to respire, right? So this paragraph describes the different mechanisms or organs of breathing. Simple. Cool. Let's go on to the human respiratory system. We have a pair of external nostrils, correct? Behind our nostril would be nasal chamber, right? So, nostrils through the nasal passage lead to nasal chamber. Nasal chamber opens into pharynx. Pharynx is common both for food and air, right? Pharynx will open into the larynx. Larynx will open into trachea. Correct? Trachea is a straight tube that extends up to the mid-thoracic cavity, and it will divide into two bronchi, right and left primary bronchi, at the level of fifth thoracic vertebra Easy peasy. Cool. Then the primary bronchi will undergo repeated division to form secondary bronchi, tertiary bronchi, and so on. Then you will have bronchioles, smaller tubes, and eventually they will end up in very thin terminal bronchioles. Okay? MCQ. Trachea, primary, secondary, tertiary bronchi, and the initial bronchioles are supported by incomplete cartilaginous ring. This is what you have to remember. This cartilage is hyaline cartilage. Cool. The terminal bronchiole gives rise to thin, irregular walled, vascularized bag-like structure called alveoli. MCQ. The wall of the alveoli are made up of squamous epithelium. Okay. So what is the lining of the remaining respiratory passage? It is pseudostratified, ciliated, columnar epithelium with goblet cells. Pseudostratified, ciliated, columnar epithelium with goblet cells. Okay. So nose will have pseudostratified, ciliated, columnar epithelium with goblet cells. The trachea, the primary, the secondary, the tertiary bronchi, all will have the same epithelium. Only the alveoli have squamous epithelium. Simple. One more important MCQ, super important. The branching network of bronchi, bronchioles and alveoli will be the lungs. If you recall in the class, I had explained when you see the building from within, we talk about staircases and elevators and lobbies and passageways. But from outside, you can only see the building. Same way. From inside, you will have a branching network of bronchi, bronchioles and alveoli. But from outside, you will have two lungs. The two lungs have to have a friction-free movement, otherwise breathing will become difficult. And hence, the two lungs are covered by a double-layered pleura with a fluid in between called as pleural fluid, which reduces the friction. Simple. There are two layers here. One layer of pleura is in close contact with the thoracic wall or thoracic lining. Inner pleural membrane is contact with the lung surface. Important MCQ that was discussed in the class. The outer layer of pleura is also called as parietal pleura, and the inner layer is also called as visceral pleura. Simple. The gap between the parietal and the visceral pleura will be called as the pleural space. Easy. Outer layer parietal, inner layer visceral. Cool. Another super important MCQ. The path starting from the external nostril up to the terminal bronchiole is called as the conducting part. You know why? Because air only passes. There is no exchange of gases taking place. The alveoli and their ducts are the respiratory or the exchange part, because this is where the exchange takes place. So this is exactly what they have re-explained. Conducting part transport air to the alveoli, clears it from foreign particles, unifies the air and brings the air to the body temperature. See, there are four functions here that you have to remember. Four functions: transport, clear, humidify and temperature. And what is the function of the exchange part? The exchange part is where oxygen and carbon dioxide diffusion will take place between the blood and the atmospheric air. Easy peasy. Now, before we move ahead, understand that larynx and esophagus are both connected to the pharynx. Getting it? The pharynx, let me put it here, the pharynx will have two openings below. One will be the opening of esophagus, other will be the opening of the larynx. When we are swallowing food, the food has to go from the mouth to the pharynx towards the esophagus, correct? It should not go to the larynx. So there has to be a mechanism that closes the larynx during swallowing of the food. This mechanism is a thin, elastic, cartilaginous flap. It is a thin, elastic cartilaginous flap called epiglottis that prevents the entry of food into the larynx. Easy, simple, cool, perfect. Acha, one more thing. The larynx is also called as the sound box because this is where we make the sound. MCQ. Larynx is called as the sound box and larynx is a cartilaginous box. Okay, it is made up of cartilage. So on this page, following MCQs have to be understood. The lining epithelium of the respiratory passages, pseudostratified ciliated columnar epithelium with goblet cells, except at the level of alveoli where it is squamous epithelium, correct? The supporting cartilages in the trachea, primary, secondary bronchi are hyaline cartilages, simple. And the cartilage of the epiglottis is elastic cartilage. Fantastic. Do not forget the parietal and the visceral pleura, the pleural space in between, and its role in reducing friction. Simple. Also, do not forget the role of the conducting part. Perfect. Moving on. The lungs are located inside the thoracic chamber. Simple. MCQ. It is anatomically an airtight chamber. How do you make this anatomically airtight chamber? You will have 12 thoracic vertebrae behind. Ventrally you will have sternum and laterally you will have ribs. I believe you remember there are 12 thoracic vertebra plus 1 sternum plus you will have 24 ribs. Correct? So the thoracic cage or the thoracic chamber has a total of 24 plus 1, 25 plus 12, 37 bones. Easy. What about the lower side? At the lower side, you have the diaphragm, the dome-shaped diaphragm. The setup is such that, super important MCQ, the setup is such that, the anatomical setup is such that any change in the volume of the thoracic cavity will be reflected in the lung cavity. If you increase the volume of the thorax, it will lead to increase in the volume of lungs. If you decrease the volume of thorax, it will lead to decrease in the volume of the lung. That is what you have to understand. Super simple. Following steps are involved in respiration. 1, breathing, also called as pulmonary ventilation. You take in air, you throw out carbon dioxide. Diffusion of gases, transport, diffusion between blood and tissues, and utilization of oxygen and release of carbon dioxide. I believe this is pretty simple and logical, but if you still have doubt, let me know. Let's understand how is the air taken in and thrown out. Okay? Step by step. First, breathing involves 2 things. Taking air in, throwing air out. Taking air in is known as inspiration. Atmospheric air is drawn in. Now, if you want to take the air in, the pressure inside the lungs should be lesser than the atmospheric pressure, right? Only then the air will go in. Right now, inspiration can occur only if the pressure inside the lung, the intrapulmonary pressure, is lesser than the atmospheric pressure. In other words, there has to be a negative pressure in the lungs with respect to atmospheric pressure, only then the air will go in. When will air come out? Expiration is when alveolar air is released out. Expiration can take place only when the intrapulmonary pressure is higher than the atmospheric pressure. Easy. Clear? So, how is it that you create a pressure gradient to move the air in and out of the lung? How do you do that? Well, simple. You have special muscles for it. The muscles are diaphragm and a special set of muscle located between your ribs. In bio, ribs are called costa. So the ribs muscles between the ribs will be called as intercostal muscles. And there are two of them, the external intercostal muscle and the internal intercostal muscle. Now in this case, you will remember the mechanism by using this thing. E is for I and I is for E. What does it mean? If you're using external intercostal, you will cause inhalation. And if you're using the internal intercostal muscle, you will call exhalation. External causes inhalation and internal causes exhalation. Easy peasy. Cool. Fantastic. So diaphragm and a special set of muscle called as the external intercostal muscles between the ribs help in generation of this gradient. So how do you breathe in? Inspiration is by contraction of diaphragm. The diaphragm normally looks like this. It is dome shaped. It's a dome shaped muscle. When it contracts, it becomes more flatter. Now because it becomes flatter, it literally pulls the lungs in the downward direction. Now in our book, this is how it has been explained. The contraction of the diaphragm increases the volume of the thoracic chamber in the antero-posterior axis. Super important, must know. Contraction of the diaphragm increases the volume of the thoracic chamber in the antero-posterior axis. At the same time, the external intercostal muscles contract. It lifts the ribs and the sternum. And what will it do? It will increase the volume of the thoracic chamber in the dorsoventral axis. Super important. In the dorsoventral axis. So diaphragm does it in the anteroposterior and intercostal muscle in the dorsoventral. Both increase the thoracic volume. But remember this line here, this one here, the anatomical setup is such that any change in the volume of the thoracic cavity will be reflected in the lungs, correct? So if by using diaphragm and external intercostal, you are increasing the thoracic volume, there will be an increase in the pulmonary volume also. But you are smart, you know this, volume is inversely proportional to the pressure. So if volume is increasing, it literally means the pressure should be decreasing, correct? Excellent. So an increase in the pulmonary volume decreases the intrapulmonary pressure. And as the pressure becomes less than the atmospheric pressure, air will move from high pressure atmosphere outside to low pressure lungs inside. That leads to inhalation. Simple. And the reverse will happen during exhalation. Relaxation of the diaphragm will take place. The intercostal muscles will will relax, and that will kind of bring the sternum, the ribs, the diaphragm back to the normal position. The thoracic volume decreases, pulmonary volume decreases. But when volume decreases, when volume decreases, the pressure will increase, correct, right? So when volume decreases, pressure will increase. That is exactly what we are talking about here. Increase in the intrapulmonary pressure. And as it becomes more than the atmospheric pressure, expulsion of air will take place. This is expiration. Perfect. Now, note, we have the ability to increase the strength of inspiration and expiration with the help of additional muscles, like in abdomen. Did you notice in this entire description, we did not talk about internal intercostal muscle. Normal respiration does not require internal intercostal muscle to work. Simple. Acha. How many times do you breathe in? A typical healthy human will breathe 12 to 16 times per minute. And how do you estimate the volume of air moving in and moving out? We use a technique, we use an instrument called a spirometer. And using spirometer, we can understand how much is the patient breathing in and breathing out. and that helps in assessment, clinical assessment of pulmonary functions. Cool. Now, for the time being, we are going to skip the respiratory volumes and capacities and directly jump to exchange of gases. So, whenever you are also studying, see to it that you skip this part first, come to exchange of gases, finish this part and go back. Okay? Now, exchange of gases. Where does exchange of gas take place? Actually, exchange of gas takes place at two places. Two places. First is alveoli, between alveolar air and the blood vessel, correct? Second site of exchange of gas is at the level of tissue, between blood and the tissue. But we are more interested in the alveoli. We will talk more about alveoli and we will then talk about the tissue. Remember, this entire process is diffusion. Diffusion is based on concentration or pressure gradient. Clear? 1. Diffusion is based on pressure or concentration gradient. 2. Diffusion is also based on solubility of gases. Correct? And 3. It is also based on the thickness of the membranes involved in diffusion. So, our textbook gives us 3 points on which diffusion will be based on. 1. Pressure gradient or concentration gradient. 2. Solubility of gases. And 3 is the thickness of the membrane. Now, how do you measure the pressure gradient? The pressure contributed by each gas in a mixture is called partial pressure. I believe you know this. So for oxygen, we'll call it PO2, and for carbon dioxide, we'll call it PCO2. We are only interested in these two gases. Others are irrelevant as of now. Now, understand, why is the air with oxygen coming inside the lungs? Why not oxygen from the lungs go? Array, simple. Concentration gradient. Look at this. How much oxygen is present in the atmospheric air? 159. Correct? Look at the table and that should be easy to solve. 159. That much is the PO2 in atmosphere. But look at alveoli. It's only 104, na? So logic. Oxygen has to come from the atmosphere into the alveoli. Now, alveoli will receive which kind of blood? Deoxygenated blood. Look at the deoxygenated blood. It only has 40 millimeters of Hg, oxygen. So, logically, oxygen will move from higher concentration alveoli to lower concentration wala deoxygenated blood. Clear? And that is the reason why the oxygen in the blood has increased from 40 to 95. Simple. When this oxygenated blood reaches the tissue, look at the oxygen in the tissue, only 40. How will the oxygen move? From higher concentrated oxygenated blood to the lower concentrated tissue. Can you see the gradient happening here? Atmospheric air 156 mmHg and alveoli only 104. So oxygen has to come in. Deoxygenated blood only has 40. So oxygen will go in, right? And that is why the oxygenated blood has higher oxygen of 95. But the tissue only has 40. So it will give away oxygen. See, everything here is happening along the concentration gradient. Acha, what is happening in the tissue? Look at what is happening in the tissue. The tissue is producing carbon dioxide. How much is the carbon dioxide? 45. When this oxygenated blood comes to the tissue and as it is giving away oxygen, look at the carbon dioxide the oxygenated blood has. The arterial blood or the oxygenated blood only has 40 mmHg carbon dioxide. Tissue has how much? 45. Don't you think oxygen will come from the tissue to the blood? Right, na? And that's why the deoxygenated blood will have PCO2 of 45. Look at alveoli. How much carbon dioxide does it have? 40. But deoxygenated blood has 45. Don't you think there is a concentration gradient? Carbon dioxide will automatically move from higher concentrated deoxygenated blood to lower concentration alveoli. And how much is the carbon dioxide in the atmosphere? Only 0.3. Don't you think carbon dioxide will automatically move out along the gradient? Was there any reaction taking place? No. Was there any energy involved? No. Was there any chemistry? No. This is not a chemistry. This is not any reaction. This is a simple physical process. Things are moving from a region of higher concentration to a region of lower concentration. And that is exactly what they have done here. They have given you how much is the PO2, how much is the PCO2, and why oxygen will go towards and why carbon dioxide will come out. Easy peasy. This is a special important diagram based question. Please see to it that you do it properly. Clear? So as you read this, you will realize the same. If you look at the table, the data given in the table clearly indicates a concentration gradient for oxygen from the alveoli to the blood and from the blood to the tissue. Similarly, there is a gradient for carbon dioxide in the opposite direction from tissue to the blood and from the blood to the alveoli. Correct? Now, if you notice, look at the gradient for oxygen, 95 and 40, right now. Such big gradients. And for carbon dioxide, it is 40-45. Why such bhedbhav? Why such difference? Array, what happens, the solubility of carbon dioxide is 20 to 25 times higher than that of oxygen. Because carbon dioxide is much more soluble. Even a small concentration gradient is enough to transport it. Getting it? Solubility of carbon dioxide is 20 to 25 times higher than that of oxygen. The amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure. Oxygen 1 mm difference, less will cross the membrane. Carbon dioxide 1 mm difference, concentration difference, more will pass. Simple. Now what exactly is this diffusion membrane made up of? Where is this diffusion membrane? Between the alveoli and the blood. And it is made up of 3 major layers. What are the 3 major layers? First, it's the squamous epithelium of the alveoli. Getting it? Second, it's the squamous epithelium of the blood vessel. You know what is the epithelium of blood vessel called as? Endothelium, which is squamous epithelium. Alveoli, we'll call it squamous epithelium of alveoli. Correct? Thin squamous epithelium. And between them is a combination of basement membrane. The basement membrane of the alveoli, squamous epithelium, and the basement membrane of endothelium. Are you getting this? And this combo basement membrane in our textbook is described as the basement substance. Simple. So the 3 layers are thin squamous epithelium of the alveoli, the endothelium of the alveolar capillaries, and the basement substance. Don't write basement membrane, please write basement substance. Now when I draw it, when I describe it, looks like a very thick membrane. The total thickness is less than 1 millimeter. So the gradient, the solubility and the thickness of the membrane is such that all factors in our body favor the diffusion of O2 from the alveoli to tissue and carbon dioxide from the tissue to the alveoli. I believe that is easy to understand. This brings us to the transport of respiratory gases. Now, as far as transport of respiratory gases is concerned, for the time being, we are going to use a diagram to understand this. Okay, and I am going to add a diagram at the end. Okay, so that you can understand this better. Okay, so here it is. Okay, I'm going to divide my page into This is where the lungs are. This is where the tissue is. Okay? And we will talk about transport of oxygen and transport of carbon dioxide. So this is how things are actually happening. Oxygen has entered our alveoli in the lungs. Getting it? This oxygen will first come into the plasma, and 3% of it will remain dissolved in the plasma itself. %3 is dissolved. Simple. The other 97% combines with a molecule called as hemoglobin and forms HbO2. That's oxyhemoglobin. One hemoglobin molecule can bind up to 4 oxygen molecules. One Hb can bind to 4 O2. And this HbO2 reaches your tissue. If needed, the oxygen is released and hemoglobin becomes free again. Easy, simple to understand. This is 97% oxyhemoglobin. This oxygen enters into the tissue and undergoes metabolism, and what is formed is carbon dioxide, right? Carbon dioxide now comes into the plasma. Let me use a different color for carbon dioxide so things don't get messy. Okay, let me use the blue color. Okay, what is made? Carbon dioxide. Carbon dioxide comes in the plasma. Carbon dioxide has the following options. Okay, option number 1, this hemoglobin molecule here can combine with my carbon dioxide to form something called as carbaminohemoglobin. This carbaminohemoglobin will go towards our lungs and it will kind of dissociate to form hemoglobin and carbon dioxide. This carbon dioxide will be exhaled. 20 to 25% of carbon dioxide travels as carbaminohemoglobin. Easy. Second method of transport is in the dissolved format. Okay, so second is dissolved. This is about 7%. Simple. But the third mechanism is super important. Let's understand this. Now this is what happens. The carbon dioxide enters into our RBC. You know what is there inside RBC? RBC has a special enzyme called as carbonic anhydrase. Here, water molecules plus carbon dioxide form H2CO3, which immediately splits into H plus and HCO3 minus. Simple. Our RBC doesn't like HCO3 minus, it throws it out. Now this creates a problem because when you throw HCO3 minus out, you're actually throwing a negative charge. If you throw out a negative charge, you have to take in a negative charge. Some negative charge has to come in. Now who's that negative charge? The sodium chloride that we eat in our food dissociates into chloride and sodium. Getting it? This chloride enters into the cell in exchange of HCO3 minus, and Na combines with HCO3 minus to form NaHCO3. Getting the idea? 70% of carbon dioxide travels as bicarbonate. Easy, cool, nice. Now once this RBC reaches the reaches the lungs, the alveolar capillaries, reverse will happen. How will reverse happen? The NaHCO3 will split into HCO3 minus and Na plus. HCO3 minus will be taken in, and what will be thrown out? Chloride will be thrown out. Getting it? And again this will form NaCl. Correct. H plus was already present here. See, H plus was already here. That H plus will now combine with HCO3 minus to form H2CO3. Carbonic anhydrase will split it into H2O and CO2, and this CO2 will be released out. Remember, this reaction can only happen inside the RBC because RBCs contain a high concentration of which enzyme? Carbonic anhydrase. It's not like there is no carbonic anhydrase in the plasma, but plasma is almost negligible. Okay, if you remember this flow chart, this entire page is done. The transport of oxygen, this paragraph and this paragraph, everything gets covered in that one particular flow chart. What is not covered is this particular graph. Okay, let me help you understand this graph also. So here on the last page we had made the transport of gases. Let's understand the experiment for HbO2 dissociation curve. Okay, now scientists did a series of experiments in which they took test tubes. Okay, multiple test tubes, of course, right? In my example, I am taking about let's say 6 of them. Let's make it 10. Okay, 2, 4, 6, 8, 10, right? In each one of them they put known amount of hemoglobin. Okay, all of them had, let's say, 5 grams of hemoglobin. Okay, shabbily drawn, but get the point, please. Okay, and they added increasing concentration of oxygen. So here, let's say 10% oxygen, 20%, 30%, 40% oxygen in this one, and so on and so forth till 100% oxygen. Getting it? And what they expected was, when you draw a graph of let's say O2 provided or PO2 versus HbO2, what they expected was they will get a straight line. Correct? 10% oxygen, 10% HbO2, 20% oxygen, 20%, 30, 30, 40, 40, and so on. That is what they expected. Surprise, surprise, what they got was a completely different graph, which was a sigmoid shaped graph. Now, what is the significance of this graph? The graph simply helps us understand this. At lower concentration of oxygen, hemoglobin does not want to bind to oxygen. At lower concentration of oxygen, hemoglobin is not able to pick up enough oxygen, let's say that way. At higher concentration of oxygen, hemoglobin doesn't want to leave oxygen. But look at this part here, between this point and this point. A small change in the oxygen concentration leads to a large change in the concentration of HbO2. Getting it? Clear? And scientists were surprised at this graph. How is this graph coming? And what is the significance? So we understood from this graph one very important thing. Hemoglobin works best in this narrow range between here to here, let's say. A small change will lead to a large change in the oxyhemoglobin concentration. Getting it? So scientists were like, awesome. Experimentally, this is what we are finding. What happens in our body? When they tried to find that out, they found that the graph in our body shifts either towards the right or it shifts towards the left. Clear? The graph may shift to left or the graph may shift to right. They also notice that this shifting to right takes place in the tissue and this shifting to left takes place in the lungs. So why is the graph shifting? Let me explain, then we can look at the textbook. Let's say this is 50% saturation. Getting it? This is 50% line. Let's presume this is 50% line. This is 100% oxygen saturation. This is 50%. This is 100%. This is 50%. Look at what is happening when the graph shifts from purple to blue. At lesser oxygen concentration, 50% hemoglobin has got saturated. Getting it? I hope I'm making sense. When the graph is shifting to the left, at lesser oxygen, you get 50% saturation. But when the graph is shifting to the right, at higher O2 concentration, you get 50%. What we are trying to suggest now, what I am trying to suggest is, whenever the graph is shifting to left, uptake of O2 is easy. Lesser concentration में ज़्यादा oxygen उठाएगा. When the graph is shifting to right, release of O2 is easy. जब ग्राफ राइट शिफ्ट करेगा, ऑक्सीजन को रिलीज किया जाएगा. That's why factors that shift to right are present in tissue and factors that shift the graph to the left are present in the lungs. So what are the factors present in the tissue that shifts the graph to the right? Please note. In right, there's low O2, high PCO2. टिश्यू में ऑक्सीजन कम होता है, टिश्यू में कार्बन डाइऑक्साइड ज़्यादा होता है. कार्बन डाइऑक्साइड का मतलब एसिड. CO2 प्लस H2O गिव्स यू H2CO3 विच विल गिव यू H प्लस. बराबर. सो जहाँ पे कार्बन डाइऑक्साइड ज़्यादा, देर विल बी हाई H प्लस कॉन्सेंट्रेशन एंड टिश्यू हैज़ हायर टेम्परेचर. So the graph will shift to right and there will be dissociation of oxygen from the hemoglobin. Clear? Which is the factor shifting the curve to the left? Left means alveoli. Getting it? Left means alveoli. High O2, low CO2. Alveoli has high oxygen, low carbon dioxide. Low carbon dioxide means less acid, less H plus and lower temperature. This will favor formation of oxyhemoglobin. So always remember this. Shift to left is lungs, and lungs means uptake of O2 means formation of oxyhemoglobin, and right is equal to tissue, which means release of O2. Clear? Two important MCQs before we move ahead. Every 100 ml of oxygenated blood delivers 5 ml oxygen to the tissue under normal conditions, and every 100 ml of deoxygenated blood delivers 4 ml of carbon dioxide to the alveoli. The rest of the things have been explained. I recommend you go through this, and if you still have a doubt, drop in a message. I will really help you. The last part of this chapter deals with regulation and some disorders. Okay, so let's have a look at how the regulation takes place. Our NCERT textbook is very clear about it. Okay, there is a center in the medulla oblongata. Okay, this system is called as the respiratory rhythm center, and this respiratory rhythm center is going to control the respiration. Okay, this is primarily responsible for the regulation of breathing. Okay, how much should you breathe? Whatever is the demand of the body. Simple. Okay, so human beings have a significant ability to maintain and moderate respiratory rhythm to suit the demands of the body. And who does it? The neural system. Where is this neural system present? A specialized center is present in the medulla region of the brain. What is it called? RRC, respiratory rhythm center. Now, above this center is pons. a center in the pons is called as the pneumotaxic center. And the pneumotaxic center controls the respiratory rhythm center. How does it do that? The pneumotactic center, remember it's in the pons region of the brain, moderate the function of the respiratory rhythm center. How? Neural signals from the pneumotaxic center reduces the duration of inspiration, and by reducing the duration of inspiration it can alter the respiratory rate. Now how do these centers know when to breathe more and when to breathe less? So there has to be a mechanism to detect the needs of the body, no? So there will be two centers. One center, let's call it a receptor or a chemosensitive area, okay, which is near to the respiratory rhythm center, and then there are other centers located near the aortic arch and into the carotids, okay. Not a absolute correct diagram, but manageable, okay. So in the carotids and in the aorta you have other centers which can send message towards the respiratory rhythm center. Clear? So you will have a chemosensitive area, super important, chemosensitive area adjacent to the rhythm center. This is sensitive to carbon dioxide and H plus, okay. This is for CO2 and H plus, okay. Any increase in CO2 or H plus will activate the center, which will tell the respiratory rhythm center to make the necessary adjustment. So you can throw away the carbon dioxide, throw away the H plus. Other receptors are present in the aortic arch and the carotid artery. They can also detect carbon dioxide and H plus concentration, and they will send the message back to the respiratory rhythm center for necessary actions. Now, if you notice, all the centers that we are describing are detecting changes in the CO2 and H plus. None of them is checking oxygen. The role of oxygen in regulation of respiratory rhythm is insignificant. Simple. Moving on. Disorders of respiratory system. There are following diseases. 1. Asthma. Asthma, whatever you want to call it. It is difficulty in breathing. Now, because of the spasm or the inflammation of the bronchi and bronchioles, they are becoming narrower, okay. Inflammation is causing spasm of bronchi and bronchioles. That leads to formation of a special sound called as wheezing. So when you read this word wheezing, think of asthma. Important MCQ: asthma is an allergic disorder. Second is emphysema. Emphysema is a disease that is commonly seen in cigarette smokers. It's a chronic disease, which means it takes time to occur. It doesn't happen like one puff of cigarette smoke and you get a disease. It takes months or years to happen, in which the alveolar walls are damaged. Remember, alveoli is the place where exchange of gases takes place. If the alveolar wall is damaged, the respiratory surface area is decreased, right? And that's how the patient will have hypoxia. You know what is hypoxia? Hypo means less, oxia means oxygen. Okay, these people will also show cyanosis. Cyanosis means bluish discoloration of the skin of their nails. Cool. The third disease in our syllabus is occupational lung diseases or respiratory disorders. Like in certain industries involving grinding or stone breaking, the dust particle is so fine that it goes right into the alveoli. And the defense mechanism of the body cannot cope up with this situation. The WBCs are thinking, where the hell is this dust particle coming from? Let me try to destroy it. But in the process of trying to destroy the dust particles, the WBCs end up harming our own lungs. Getting it? One time event, maybe not much of a problem, but long exposure will lead to repeated inflammation. Repeated inflammation will lead to fibrosis. Fibrosis means proliferation of fibrous connective tissue, and that leads to serious lung damage. So how can you protect these workers? Give them protective masks. Simple. Now the only thing remaining in this chapter, if you recall, is this spirometry. A spirometry is basically it's an experiment to find out how much is the patient breathing in and breathing out. Okay, so let this be the volume of air a patient is breathing in and breathing out. Now this is what happens. You take a deep breath in spirometry na, upstroke is equal to inhalation and downstroke is equal to exhalation. Upstroke is inhalation, downstroke is exhalation. Okay, so when you ask the patient to breathe in, breathe out, breathe in, breathe out, breathe in, breathe out, upstroke inhalation, downstroke exhalation, you notice that these waves, they look like tides coming in and going out. That's why normal breathing in and normal breathing out is called as tidal volume. The volume of air inspired or expired during a normal respiration. How much is this volume? 500 ml. In 1 minute, how much it will be? 500 into, remember, 12 to 16 times per minute, no? So 6000 to 8000 ml of air per minute. Simple. Now, next, you ask the patient to take a normal breath in. So patient takes a normal breath in till here, correct? Let me draw this further. So we are here. Then you ask the patient, breathe in as much as possible. So the patient breathes in max and back to normal. Getting it? This, whatever blue part I have drawn, is called as inspiratory reserve volume. This much air the patient can inspire by forceful inspiration. How much is that? 2500 to 3000 ml. Then you ask the patient, after normal exhalation, exhale as much as you can, and the patient exhales as much as he can. This is called as expiratory reserve volume. So this is IRV and ERV. Simple. What is the expiratory reserve volume then? The air expired after forceful expiration. It is 1000 to 1100 ml. Clear? And if you notice, even after full exhalation, a certain amount of air still remains locked inside. Certain volume of air still remains locked inside. This cannot be measured by spirometry. The volume of air remaining inside the lungs even after forcible expiration is called residual volume. It is 1100 to 1200. Then you can just keep adding the volumes to get various capacities. Inspiratory capacity is tidal volume plus IRV. On this graph, this much. Right? Can I say after normal exhalation here? After normal exhalation, what is the maximum air you can breathe in? That is your inspiratory capacity. See, this is what they have written. The total volume of air a person can inhale or inspire after normal expiration. Simple. Reverse would be expiratory capacity, tidal volume plus ERV. On this graph, it will be this one. From here to here. Getting it? TV plus ERV. How much amount of air can a patient breathe out after normal inhalation? After normal inhalation, you are here. After maximum exhalation, you are here. Okay? Simple. What is the amount of air that remains normally inside the lungs? Functional residual capacity, ERV plus RV. Simple. Vital capacity is a total of ERV, TV plus IRV, whereas total lung capacity is a combination of all of them. Easy peasy. Now you have to remember the description: volume of air that remains in the lungs after normal exhalation, maximum volume of air a person can breathe it after forced expiration. See, these are potential MCQs. Do this much and you should be able to get almost all the questions correct from this particular chapter. Okay? Be calm, composed, relaxed when you're doing this chapter. This chapter is super duper simple. You should be able to get full marks. Let me know if you have any doubts. I will be more than happy to help you. Till the next time we meet, this is Dr. Avinash signing off for now. Take care, keep studying and keep rocking. Bye bye.